Right. So in this video, I'm just going to be talking about different terminologies that you might encounter, especially in this part of the course where we talk about transcription and translation or lack operon. And a lot of these courses, you will also encounter them in other biology classes. So I think it's good to be familiarized with what they are. So yeah, let's just get started. DNA. DNA is just the whole genetic makeup of the organism. When professors talk about DNA, they just literally mean like your whole genome or your whole DNA sequence. And then gene. Gene is just the specific section of the DNA. Because that specific section of the DNA, it has information to make something or to make a protein or an RNA. And in a way, if you visualize what I mean, what I'm saying, if this is your DNA, the gene is only like this section over here, right? Now, this section over here, when expressed or when read through, we will make the protein or an RNA. That process is called gene expression because we're reading it, taking it apart, and making something out of it. And you might be familiar with what those terms are. Gene expression is divided into two, which is transcription and translation. In transcription, the most basic way I can think of is turning DNA into RNA. And then translation is just turning RNA into a protein or a polypeptide. My trick here to remember what the difference are between gene, translation, transcription is that gene is the recipe, and then transcription is taking notes or making a copy of it in another paper, like paraphrasing everything. And then translation is just cooking the recipe to make the food. Because recipe is like the instruction, and then taking notes is just it's just transcribing or like paraphrasing everything else. And then the food that is made from cooking this is the protein or the RNA. All right, so let's get back here. Transcription. In the more formal sense, transcription is just the process of copying the gene's DNA sequence into an RNA sequence. The RNA sequence could be mRNA or messenger RNA. It could also be ribosomal RNA, tRNA, miRNA, SN. There's a lot of RNAs out there, but the main point here is that it's just copying the gene's DNA sequence into the RNA sequence. That is the main point here. Now, in transcription, you will see these words called template strand, coding strand, and RNA strand. Let's start with template strand. Template strand is also known as non-coding strand. Remember this, it is one of the most confusing part of the of this course. When we talk about template strand, it is the strand that the RNA polymerase uses to make the mRNA. Because remember, RNA polymerase makes an RNA sequence, right? And then that sequence is complementary to the template strand. And then the template strand is also complementary to the coding strand. It is also a DNA sequence. And what I mean by that is that it uses thymines or your T's. Now, coding strand. Coding strand is also known as non-template strand. These are interchangeable words. Template strand is also known as non-coding strand. And then coding strands is also known as non-template strands. These are interchangeable words, so sometimes you might see in the exam, if I have this non-template strand, what is the template strand? Right? Or if I have this non-coding strand, what is the coding strand? So just be familiar with it. Like Template strand goes with non-coding strand, and then coding strand goes with non-template strand. All right, so coding strand. Coding strand is complementary to the template strand in the same way that template strand is complementary to the coding strand. It is also identical to the RNA strand that will be produced when you transcribe. So the only exception is that the thymines are exchanged with uracils, and it is still a DNA sequence because it still uses thymines. Now, RNA strand. RNA strand, when we talk in transcription, when we talk about RNA strand, we normally talk about the messenger RNA or the mRNA. Because in transcription, we're basically just making the mRNA normally. Um, RNA strand, it is complementary to the template strand. It is identical to the coding strand in a way that everything is the same except for the T and the U's. And then it is an RNA sequence because we use uracil instead of thymine. All right, so maybe this will, if you're still getting confused, maybe this will make sense if we do an example over here. All right. The first example is that let's say we have a given template strand. Template strand is given. We have to write the coding strand and the mRNA strand. Now remember, when you're making your strands, you have to make sure to put your polarities or your appropriate labels. And that is your 3 primes and 5 primes, right? Coding strand. Coding strand is just the opposite or the complementary strand of your DNA sequence, right? So if I'm gonna make this, if this is my template strand, complementary to that is my coding strand. So that means complementary to this would be TAG. Remember, these are important. Your labels, like your polarities, these are very important to include, especially in your exam. You will always want to make sure that you write the actual polarities of it. Now, mRNA strand. Let's say I didn't write this coding strand. Let's make this white. When we're making the mRNA strand, it has to be complementary to the template strand, just like what I mentioned here. RNA strand is complementary to the template strand. So that means if I'm gonna make this, it will look something like this. Right? Now, let's compare this to what I wrote before, to the coding strand. If you look at this, coding the mRNA strand and the coding strand look similar, right? Except for the T's and the U's. Like everything is the same, your AGA, CCC. So, in a sense, the mRNA strand and the coding strand are the same, except for the thymines and the uracils. But they're both of them are complementary to the template strand. All right, let's do another example. Let's say we're given the coding strand instead, and we have to write the template and the mRNA. So if my coding strand is 5 prime to 3 prime, remember it has to be anti-parallel and you have to put your appropriate labels. So this is my template strand because remember, the coding strand is complementary to the template strand, right? Now, we also have to make the mRNA strand, right? You can pause the video and try to make it yourself and then go back here and see if you got the same thing. So, like I said before, the mRNA strand looks similar to the coding strand, even with the polarities like 3' to 5' or 5' to 3'. But it is also complementary to the template strand. So if we apply that here, I can just copy my coding strand, which is something like this. You can see that my mRNA strand is similar to the coding strand, except for the thymines and the uracils. And then it is also complementary to the template strand, with it being 3' to 5', and then the mRNA strand being 5' to 3'. So it is it is really good practice for you to try out in both directions, like starting with a 3' to 5' sequence or 5' to 3' sequence. Now, try pausing the video and do this by yourself and then come back here and see the answers. I will put the answers over here. All right, so this is just the few examples that I can give out. Um, yeah, this is basically the whole video about the terminologies and some examples. Kenzie also made a video of hers. I would suggest watching it also. You might get different perspectives from listening to her video as well. And yeah, that's it for this video and hope this actually helped you. Thank you.